Hello everyone. So today we'll be going into the regulation of transcription, translation and protein synthesis. So yeah, that's what we're going through today. So let's get into it without wasting time. Transcription is the process by which RNA is formed, and translation is the process whereby by which proteins are formed. So now these processes have strict regulation so as to control the expressions of genes, because the expressions of genes is the synthesis of proteins. So now this has to be regulated. So now there are different areas that is actually regulated, as from the DNA to the RNA, which is transcription, is a regulated process. Then from the RNA transcript to the mRNA, there's also post-transcriptional modification that takes place, so it's also regulated at that point. And then the RNA transport from the nucleus into the cytoplasm is also regulated. And then the translation from mRNA to proteins is a regulated process. And then the mRNA that's already in the cytoplasm can also be regulated. They can also be regulated whether they are degraded and then they become inactive mRNA. And now the proteins that are synthesized can either be inactive proteins or active proteins. So this is how the biological system regulates the formation of proteins. So now we'll be considering few regulatory process by endogenous factors, and then we'll be going to the transcription factor, the chromatin modification, the RNA processing, and the DNA methylation. Now transcription factors are either repressors or activators that bind to the DNA sequence, inhibit or repress transcription from the DNA to the RNA, or they can affect the activity of the RNA polymerase, while the activators will actually increase the transcription of the particular gene. Now for chromatin modification, now chromatin is made up of histone and DNA. So now this histone there can be methylated or acetylated, and then when they are methylated or acetylated, it exposes the DNA more or makes it more accessible to the RNA polymerase so that it can be effectively transcribed into the RNA. The RNA processing, the RNA transcript is processed into the mRNA. But now the RNA is made up of introns and exons. RNA processing involves the removal of the introns and leaves only extrons. But now it will be regulated at this process, in which that some introns will actually be left when the mRNA is being formed. So now when it comes to translation, these introns will cause some problems or will bring about reducing the translation of that mRNA into proteins. Now, and then the last one we have is DNA methylation. Now a particular part of the DNA is methylated by the methyl group. A methyl group is attached to a particular segment of the DNA. Now it alters the property of that segment but not the entire sequence. So we take, for example, we have a GC 8C GC sequence. Now the G is methylated. It only changes that segment but does not changes the entire sequence. The sequence remains the same. When a promoter site is methylated, it represses the transcription of the DNA. Now also consider some exogenous factors that actually cause dysregulation. First one we're looking at here is rifampicin. Now rifampicin binds the beta subunit of the RNA polymerase of prokaryotes. So that means this rifampicin does not affect eukaryotes but only prokaryotes. It causes inhibition of transcription in prokaryotes. This rifampicin is a powerful drug that can be used against bacterial infections. An example of bacterial infections can be used against is tuberculosis and leprosy. Now the next exogenous factor we're considering is actinomycin D. So now it acts to inhibit or binds to the RNA polymerase of both prokaryotes and eukaryotes this time, and then inhibits or affects transcription from DNA to RNA. So now how it does this is by intercalation. It intercalates in the DNA and distorts the structure of the DNA. So now it acts between two successive GC base pair. It's a concentration dependent action. If there's high concentration of it, it will cause more deformation of the DNA. And then acridine also functions in a similar way. Now acridine, we considered it under our toxicology course, that it also performs intercalation of the DNA. Now we are coming to regulation of transcription with Operon. Now, in prokaryotes, genes that they evolved in related functions are regulated as a single unit. Now they are organized as operons and then they are regulated as a single unit. So now there are different mechanisms by which these operons are actually regulated. Now the first one here is repressors and activators. Now these repressors and activators can bind to specific DNA sequences near the operon and affect the binding of the RNA polymerase to form the RNA. Now these repressors and activators are actually transcription factors, just like the transcription factors we considered under endogenous factors that regulate RNA synthesis. Next, we have the inducers and corepressors. Now these inducers and corepressors, they bind to repressors and activators to alter their ability. So now, for example, we have lactose as an inducer in the lac operon in E. coli, and now the repressor there is lac repressor. So now this lactose binds to this lac repressor to alter its ability and prevents it from binding to the promoter site on the lac operon in the Escherichia coli in the E. coli. Now we have the alternative sigma factors that can initiate transcription by binding to different promoters in the operon. Now this operon may have different promoters, and it can bind to these different promoters to initiate transcription. It's used to regulate transcription in response to different environmental conditions. And the last one we have is attenuation. Now attenuation is a mechanism of transcriptional regulation where the mRNA that is being formed, a specific secondary structure of the mRNA is formed, and that causes termination of the transcription from the DNA to the mRNA. And this is used to regulate transcription in relation to environmental conditions. That is all for this video. So yeah, thank you, and don't forget to click the subscribe button. Thank you.